Chapter 3 Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker 1969, wherein he suggested the five kingdom classification: Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of plant kingdom has changed over time. Fungi and members of Monera and Protista cell walls have now been excluded from Plantae, though earlier classifications placed them in the same kingdom. So, the cyanobacteria that also referred to as blue-green algae and not algae anymore. In this chapter, we will describe algae, bryophyta, pteridophyta, gymnosperms and angiosperms under Plantae. Let us look at the classification under within angiosperms to understand some of the concerns that influence the classification system. The earliest system of classification used only gross superficial morphological characters such as habit, color, number, and shape of leaves. Example, they were based mainly on vegetative characters or in androecium structure. Such system was artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification systems developed, which were based on artificial affinities among the organisms and considered not only the external features but also internal features, anatomy, embryology, physiochemistry. Such a classification for flowering plants were given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationships in the various organisms are acceptable, as organisms belong to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. This becomes more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Numbers and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters are being considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy that uses chemical constituents of plants to resolve confusions, are also used by taxonomists these days. 3.1 Algae. Algae are a chlorophyll-bearing simple, thalloid, autotrophic and largely aquatic organisms. They occur in a variety of other habitats, moist stones, soils and woods. Some of them also occur in association with fungi and animals. The form and size of algae is highly variable, ranging from colonial forms like volvox and the filamentous forms like Eulothrix and Spilogyra. A few of marine forms such as scalps form a massive plant bodies. The algae reproductive by vegetative, asexual and sexual methods. Vegetative reproduction is by fragmentation. Each fragments develop into a thallus. Asexual reproduction is by production of few types of spores, the most common being the zoospores. They are flagellated and on germination gives rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix or non-flagellated, non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudonia termed as angiosperms. Fusion between one large non-motile female gamete and a smaller motile male gamete is termed as oogamous. Example, volvox and fucus. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae throughout photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are paramount importance as primary producers of energy-rich compounds which form the basis of food cycle of all aquatic animals. Many species of Porphyra, Laminaria and Sargassum are among the 70 species of marine algae used as food. Certain marine brown-red algae produce a large amount of hydrocolloids, example algin and carrageen. which are useful commercially. Agar, one of the commercial products obtained from Geladium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Cholera, a unicellular algae rich in protein, is used as food supplements even in space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. 3.1.1 Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be dicot, plate-shaped, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in form of oil droplets. Green algae usually have a rigid cell wall made up of inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagelated zoospores produced in zoosporgia. The sexual reproduction shows considerable variation in the type of formation of sex cells and in may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Spirogyra, and Chara. Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous forms to profusely branched forms as represented by caps, which may be reached in a height of 100 meters. They possess chlorophyll A, C, carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is stored as a complex carbohydrates which may be in the form of laminarian or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. The protoplast contain in addition into plastids a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stripe, and a leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in the most brown algae is by biflagellate zoospore that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the ooganium. The gamete and pyroform bear two laterally attached flagella. The common form are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. 3.1.3 Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of red pigment R-phycoerythrin in their body. Majority of the red algae are marine with greater concentrations formed in warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in oceans where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have a complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by a non-motile gamete. Sexual reproduction in oogamous and accompanied by a complex post-fertilization development. The common members are Polysiphonia, Porphyra, Glacialia, and Geladium. 3.2 Bryophytes. Bryophytes include the various mosses and liverworts. They are found commonly growing in moist shaded areas in the hills. Bryophytes are also called amphibians of the plant kingdom because their plant can live in soil but are dependent on water for sexual reproduction. They occur in damp, humid, and shady localities. They play an important role in the plant succession on bare rocks or soils. The plant body of bryophytes is mostly differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stems, or leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of the bryophytes is haploid. It produces gametes, hence it is called as gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is antheridium. They produce biflagellate anthrazoids. The female sex organ called archegonium is flower-shaped and produces a single egg. The anthrazoids are released into water where they come in contact with archegonium. An anthrazoid fuses with the egg to produce the zygote. Zygote does not undergo reduction division immediately. They produce a multicellular body called sporophyte. The sporophyte is not a free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum, a moss, provide peat that have long been used as fuel and as packing material for transshipment of living materials because of their capacity to hold water. Mosses along with lichens are the first organisms to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. 3.2.1 Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy grounds, damp soils, bark of trees, and deep in woods. The plant of the liverworts is thalloid, example Marchantia. The thallus is dorsiventral and closely appressed to the substrate. The leaf members have tiny leaves-like appendages in the two rows of the stem-like structures. Asexual reproduction in the liverworts takes place by fragmentation of thalli or by formation of specialized structures called gammae. Gammae are green multicellular asexual buds which develop in small receptacles called gammae cups located on the thalli. The gammae become detached to the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or different thalli. The sporophyte is differentiated into food, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophyte. 3.2.2 Mosses. The predominant stage of the life cycle of a moss is the gametophyte, which consists of two stages. The first stage is protonema stage, which develops directly from the spore. It is a creeping green branched and frequently filamentous stage. The second stage is a leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender, axis-bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced as apex of the leafy shoots. After fertilization, the zygote develops into sporophyte consisting of food, seta, and a capsule. The sporophyte in mosses is more elaborate than that of liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. 3.3 Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these tissues in Chapter 6. The pteridophytes are found in cool, damp, shady places through some flourish well in the sandy soil conditions. You may recall that bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte, which is differentiated into two roots, stems, and leaves. This organ possesses well-differentiated vascular tissues. The leaves in pteridophytes are small, as in Selaginella, or large, as in ferns. The sporophyte bears sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form a distinct compact structure called strobilae or cones. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous small but multicellular free-living, most photosynthetic thalloid gametophyte called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilizations, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophyte to bear a male and a female sex organ called antheridia and archegonia respectively. Water is required for transfer of anthrazoids, the male gamete released from the antheridia to the mouth of archegonia. Fusion of male and female with the egg present in the archegonium result in the formation of a zygote. Zygote thereafter produces a multicellular well-differentiated sporophyte, which is dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are similar kinds, such as plants called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro and microspores, are known as heterosporous. The megaspores and microspores germinate and give rise to a female and a male gametophyte respectively. The female gametophyte in the plant are retained on the parent sporophyte for variable period. The development of the zygote into young embryos takes place within the female gametophyte. This event is the precursor to the seed habitat, considered as an important step in evolution. The pteridophytes are further classified into four classes: Coelopsida, Lycopsida, Spinopsida, and Pteropsida. 3.4 Gymnosperms. The gymnosperms are plants in which the ovules are not enclosed by an ovary wall and remain exposed both before and after fertilization. The seeds that develop post-fertilization are not covered, that is, they are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. One of the gymnosperms, the giant redwood tree, Sequoia, is one of the tallest tree species. The roots of the generally taproots. Roots in some genera have fungal association in with form of mycorrhiza, which in some cases are specialized roots called coralloid roots and associated with nitrogen fixation cyanobacteria. The stems are unbranched or branched. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaves reduce to surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are worn on sporophylls, which are arranged spirally along the axis to form lacs or complex strobili or cones. The strobili, being microsporophylls and microsporangia, are called microsporogiate or male strobili. The microspores develop into a male gametophytic generation, which is highly reduced and confined to only a limited number of cells. The reduced gametophyte is called a pollen grain. The development of a pollen grain takes place within the microsporangia. The cones bearing microsporophylls with ovules or megasporangia called the macrosporangiate or a female strobili. The male and female cones or strobili may be born on the same tree. However, the cycas male cone and the megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucleus. The nucellus is protected by an envelope and the composite structure is called an ovule. The ovule are borne on megasporophylls which may be clustered to form a female cone. The megaspore mother cell divided mitotically to form four megaspores. One of the megaspores, enclosed within the megasporangium, developed into a multicellular female gametophyte that bears two or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms the male and female gametophytes do not have an independent free-living existence. They remain within the sporangia retained on the sporophyte. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tube carrying the male gamete grow towards the archegonia in the ovule and discharge their content near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and in the ovules in the seeds. These seeds are not covered. 3.5 Angiosperm. Unlike the gymnosperms, where the ovules are naked, in angiosperms or flowering plants, the pollen grains and ovules are developed in a specialized structure called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are exceptionally large group of plants occurring in a wide range of habitat. They range in size from smallest Wolffia to the tall tree Eucalyptus. They provide us with food, fodder, fuel, medicine, and several other commercially important products. They are divided into two classes, dicotyledons and monocotyledons. Summary. Plant kingdom include algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Algae are chlorophyll-bearing, simple, thalloid, autotrophic, and large aquatic organisms. Depending on the type of pigment possessed and the type of storage food, algae are classified into three classes, namely Chorophyceae, Phaeophyceae, and Rhodophyceae. Algae are usually reproduced vegetatively by fragmentation, asexually by formation of different types of spore, and sexually by the formation of gametes which may show isogamy, anisogamy, or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is the thallus-like and posted or erect and attached to the substratum by rhizoid. They possess root-like, leaf-like, and stem-like structure. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender, axis-bearing, spirally arranged leaves. The main plant body of bryophytes is gametophyte producing and is called gametophyte. It bears the male sex organs called antheridia and female sex organ called archegonia. The male and female gametes fuse to form a zygote, which produces a multicellular body called a sporophyte. It produces haploid spores. The spores germinate to form gametophyte. In pteridophytes, the main plant body is a sporophyte which differentiated into true roots, stems, and leaves. The organ possesses well-differentiated vascular tissue. The sporophytes bear sporangia which produces spores. The spores germinate to form gametophytes which require a cool, damp place to grow. The gametophytes bear a male and female sex organ called antheridia and archegonia respectively. Water is required for the transfer of male gametes to archegonium, whereas zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperms are the plants in which ovule are not enclosed by any ovary wall. After fertilization, the seed remain exposed and thereafter these plants are called naked seed plants. The gymnosperms produce microspore and megaspores which are produced in microsporangia and megasporangia borne on sporophyll. The sporophyll, microphyls and megasporophylls are arranged spirally to form a male and sex cone respectively. The pollen grains germinate and pollen tube release the male gamete into ovules where it fuses with the egg called archegonia. Following fertilization, the zygote develops into embryo and the ovules into seeds. The angiosperms are divided into two classes, the dicotyledons and the monocotyledons. Chapter over. Thank you.